Microbiology Chapter 13, Control of Microbial Growth. The concept of cleanliness is subjective and varies based on the context of use. For instance, while washing a car or vacuuming carpet makes these items cleaner by reducing the microbial load, they do not need to be as pristine as silverware, which is used for eating. Surgical instruments, in contrast, require a higher level of cleanliness due to their role in invasive procedures, necessitating sterilization to prevent infections. Thus, the level of cleanliness required is determined by the intended purpose of the items in question. Sterilizing all surfaces and items we encounter is not only impractical, but can also pose risks. Sterilization processes are often time-consuming and labor-intensive, potentially damaging the items or introducing harmful substances to users. Therefore, it is crucial to evaluate the intended application of an item when selecting an appropriate cleaning method to ensure it meets necessary cleanliness standards. The learning objective focuses on understanding the difference among disinfectants, antiseptics, and sterilants, as well as the principles behind controlling microorganisms through sterilization and disinfection. Additionally, the objectives emphasize differentiating between microorganisms across various biological safety levels and explaining the appropriate methods for handling these microbes at each level. To prevent the spread of human disease, it is essential to control microbial growth on inanimate objects known as fomites, such as doorknobs, toys, towels. The required level of cleanliness for fomite depends on its intended use and the resistance of potential pathogens to antimicrobial treatments. For instance, invasive application necessitate higher cleanliness standards compared to non-invasive ones. Additionally, neurotoxin-producing pathogens like Clostridium botulinum, which can survive in harsh conditions due to endospore formation, require extreme measures for eradications, such as high temperature and pressures, while other microbes may be effectively managed through simpler methods like washing in a laundry machine. For researchers or laboratory personnel working with pathogen, the risks associated of specific pathogens dictate the required levels of cleanliness and control. The Center for Disease Control and Prevention, CDC, and the National Institute of Health, NIH, have established 4 classification levels known as biological safety levels, or BSLs. Similar classification schemes are used by various organizations worldwide, including World Health Organization and European Union. According to the CDC, the BSL is determined by the factors such as agent's infectivity, ease of transmission, potential disease severity, and the nature of the work being conducted with the agent. Each BSL mandates a different level of biocontainment to prevent contamination and the spread of infectious agents to laboratory personnel and broader community. For instance, the lowest BSL, BSL-1, requires the least precaution as it pertains scenarios to minimal risk for microbial infections. BSL-1 agents are generally non-infectious to healthy human adults. Examples include non-pathogenic strains of bacteria like Escherichia coli or Bacillus subtilis, as well as viruses that infect animals other than humans, such as baculoviruses, which is an insect virus. Since working with BSL-1 agents represents negligible risk, minimal precautions are necessary. Laboratory workers employ standard aseptic technique and may work with these agents on open benches or tables, utilizing personal protective equipment, PPE, such as laboratory coats, goggles, gloves, as needed. Only a sink for hand washing and doors to separate the laboratory from the other building areas are required. Agents classified as BSL-2 pose moderate risk to laboratory workers and the community, typically being indigenous to the geographical area. This group includes bacteria like Staphylococcus aureus and Salmonella species, along with viruses such as hepatitis, mumps, and measles. BSL-2 laboratories necessitate Additional precaution beyond those of BSL-1, including restricted access, mandatory PPE, potentially including face shields, and use of biological safety cabinets for procedures that may aerosolize agents. These laboratories feature self-closing doors, eye wash stations, and autoclaves for sterilizing materials before use or disposal. BSL-1 laboratories may also have autoclaves. BSL-3 agent can cause severe infections through inhalation. These agents may be indigenous or exotic, meaning they originate from foreign locations, and include pathogens like Mycobacterium tuberculosis, Bacillus anthracis, West Nile virus, and human immunodeficiency virus, HIV. Due to the serious nature of infections caused by BSL-3 agents, laboratories working with them require restricted access. Laboratory personnel are under medical surveillance and may receive a vaccination for the microbe they handle. In addition to standard PPE, workers in BSL-3 laboratories must wear respirators and conduct all work within biological safety cabinets. BSL-3 facilities require hands-free sinks, eye wash stations near exits, and double self-closing locking doors at entrances. These laboratories utilize directional airflow to ensure clean air flows from uncontaminated areas to potentially contaminated areas with no recirculation allowed. BSL-4 agents are most hazardous and often fatal. These microbes are typically exotic, easily transmitted via inhalation, and cause infections for which no treatments or vaccines exist. Notable examples including Ebola virus, Marburg virus, and smallpox virus. Only a limited number of laboratories in the United States and globally are equipped to handle these agents. In addition to BSL-3 precautions, BSL-4 laboratory personnel must change clothing upon entering, shower upon exiting, and decontaminate all materials before leaving. While working, they must wear full body protective suit with designated air supplies or work within biological safety cabinets equipped with high efficiency particulate air HEPA filtration for both supply and exhaust. If wearing suits, the internal air pressure must be higher than the external pressure to prevent contaminated air from entering in case of leak. BSL-4 laboratories must be situated in separate buildings or isolated section of buildings with dedicated air supply and exhaust systems along with specific decontamination system. The BSLs are summarized in the left panel of the accompanying slide as well as in the table on the following slide. Here you can see all the different biosafety labels and some of the examples of the agents that you can handle in these laboratories. There are only a handful of BSL-4 laboratories in the entire world as shown here. Interestingly, SARS-CoV-2 is not considered a BSL-4 agent and can be handled in a BSL-3 laboratory. The video shows a working of the BSL-4 laboratory. BSL-4 labs are enigmatic in public eye, and many Hollywood blockbusters adopted the BSL-4 labs in their screenplay. Contagion is the 2011 film that, for obvious reason, has become one of the must-watch movies of 2020 amidst the COVID-19 pandemic. Jennifer L. was Dr. Mally Hextall, the scientist who helped discover the vaccine that saved humanity. In one crucial scene, she bravely injects herself with the new serum. We had to reshoot that. I had done it through a pair of tights, and the virologist said she would have to done it on bare skin. Columbia University epidemiologist Dr. Ian Lipkin discovered more than 2,500 viruses over his 40-plus year career and is a celebrity virologist. He was a scientific advisor of the movie Contagion. He helped the set designer create accurate costumes for the film's characters to wear. He opened his lab to teach the two stars a handful of key research skills and let the sound team record the sounds in the room. Lipkin lab personnel also helped to make the 3D model of the virus using real structural biology rendering software. This slide shows a comprehensive overview of microbial control methods. Below are the definition of some of the frequently used terms, which we will discuss in detail with example in the next slide. Sterilization is the process by which all living cells, spores, and acellular entities are destroyed or removed from an object. Sterilant is the chemical agent. Disinfection, killing, inhibition, or removal of disease-causing microorganisms, whereas disinfectants are agents usually chemical used for disinfection, usually used on inanimate objects, does not always sterilize as spores on a few organisms can remain. Sanitization is the reduction of microbial population to levels deemed safe by public health standards. Antisepsis is the destruction of microbes on living tissues. Antiseptics are the chemical agents applied to tissue to kill or inhibit growth of pathogens. Sterilization refers to the most rigorous protocol for microbial control, aiming for the complete eradication of all vegetative cells, endospores, and viruses from specific items or environment. This level of microbial control is essential in a laboratory, medical, manufacturing, and food industry setting, where items must be devoid of any potentially infectious agents. Sterilization can be achieved through physical methods, such as applying high heat, pressure, or employing filtration techniques, as well as through chemical means using agents known as sterilants. These sterilants are effective in eliminating all microbes and viruses, and with sufficient exposure time, they can also destroy endospores, ensuring a sterile environment. In clinical setting, maintaining aseptic technique is vital to prevent contamination of sterile surfaces. Aseptic technique encompasses a series of protocols designed to maintain sterility or asepsis, thereby preventing introduction of microbes and infectious agent into patients. If aseptic techniques are neglected during medical procedures, there is a risk of microbes entering the patient's body, potentially leading to sepsis, a serious systemic inflammatory response to infections, characterized by symptoms such as high fever, increased heart and respiratory rates, shock, and even death. Medical procedures that pose contamination risks are conducted within sterile fields, which are designated areas maintained free from all vegetative microbes, endospores, and viruses. Establishing these sterile fields involves strict protocols, including the use of sterilized materials and rigorous procedure for cleaning and applying sterilants, alongside additional measures to preserve sterility during procedures. A specific sterilization method in the food industry is known as commercial sterilization, which utilizes heat to destroy common foodborne pathogens such as Clostridium botulinum while preserving food quality. C. botulinum and its endospores are often present in soil and can contaminate crops during harvesting. In canned foods, these endospores may germinate in anaerobic conditions, leading to issues such as bulging can due to gas production. To mitigate the risk of contamination, commercial food canning protocols are designed with significant safety margin, targeting extremely high endospore population and reducing it to a safe level. For example, low and medium acid foods are heated to 121 degrees Celsius for at least 2.52 minutes, sufficient to decrease the population from 10 to the power 12 endospore to just 1. However, it is important to note that commercial sterilization does not eliminate all microbes. It specifically targets pathogens responsible for spoilage and foodborne diseases, allowing many non-pathogenic organisms to remain. As such, the term sterilization in this context may be somewhat misleading, with quasi-sterilization being more accurate descriptor. Other methods of control. Sterilization protocols are often impractical or unnecessary in various settings, leading to the use of alternative methods for reducing the microbial load on items in both clinical and non-clinical environments. Although the terms used for these methods are frequently interchangeable, it is important to recognize the significant distinctions among them. A comprehensive table highlights these differences, clarifying the appropriate application of each method. Disinfection is a process that inactivates most microbes on the surface using antimicrobial chemicals or heat, While disinfectants effectively kill many vegetative cells, they do not render items sterile, as some resilient microbes can survive. Ideal disinfectants should be fast-acting, stable, inexpensive, easy to prepare, and user-friendly. Natural disinfectants such as vinegar leverage their acidity to kill most microbes. Chemical disinfectants like chlorine bleach are commonly employed to clean surfaces such as laboratory benches and clinical areas, though they typically do not eliminate endospore, which can withstand disinfection. Antiseptics differ from disinfectants in that they are safe for use on living skin or tissues. Examples of antiseptics include hydrogen peroxide and isopropyl alcohol. The application of antiseptic is referred to as antisepsis. In addition to that desired characteristic of disinfectants, antiseptics must selectively target microorganisms and penetrate tissues without causing any damage. The specific cleaning protocol required varies depending on the items being treated. Clinical items are categorized critical, semi-critical, or non-critical. Critical items such as surgical instruments and intravenous fluids must be sterile as they penetrate sterile tissue or the bloodstream. Semi-critical items like gastrointestinal endoscope contact mucous membrane but do not penetrate tissues. They require a high level of disinfection rather than sterilization. Non-critical items, which include bed linens and stethoscopes, require cleaning but do not need extensive disinfections. Degerming is another process exemplified by hand washing, which significantly reduces microbial numbers on living tissues, typically skin, through gentle scrubbing with mild chemicals like soap. Alcohol swabs used at injection sites also serve as an example of degerming, which removes most but not all microbes from the skin surface. Sanitization involves cleansing fomites to lower microbial levels to a safe threshold for public health. In the food service industry, commercial dishwashers employ high temperatures to sanitize dishes by killing most microbes. Similarly, hospital rooms' surfaces are sanitized using chemical disinfectants to prevent disease transmission. A summarized table provides definitions, application, and agents used in controlling microbial growth, illustrating various protocols available for maintaining cleanliness and the safety in the next slide. The table shows a compilation of comprehensive protocols, definitions, application, and few agents of control. We will discuss these agents of microbial control in greater detail later in the chapter. Chemotherapy is a generic term that describes the application of chemicals to kill microorganisms. The "-cide suffix indicates an agent that kills. Cidal agents that kill are especially effective against a specific group. For example, that would include bactericides, fungicides, and viricides. The static suffix indicates growth-inhibiting agents. Static agents inhibit growth, which is especially effective against a specific group. That may include bacteriostatic, fungistatic, virustatic, etc. Although the static treatment do not kill infectious agents, but are often less toxic to humans and animals, preserving the integrity of the treated items. These treatments effectively control microbial population and can be safely incorporated into plastics, preventing microbe growth on surfaces like children's toys, food preparation, cutting boards. When applied to infections, static treatment can adequate for healthy individuals by inhibiting pathogen multiplication, allowing their immune system to eliminate the infection. The pattern of microbial death mirrors the pattern of microbial growth. The degree of microbial control can be evaluated using microbial death curve to describe the progress of effectiveness of a particular protocol. When exposed to a particular microbial control protocol, a fixed percentage of microbes within the population will die. Because the rate of killing remains constant, even when the population size varies, the percentage killed is more useful information than the absolute number of microbes killed. Death curves are often plotted as semi-log plots, just like the microbial growth curves, because the reduction in microorganisms is typically logarithmic, as shown in the slide figure. The decimal reduction time, or the D value, is the amount of time that it takes for a specific protocol to produce one order magnitude decrease in the number of organisms or the death of 90% of the population. Is called the decimal reduction time, DRT, or the D value. The Z value represents the temperature change, increase in degree Celsius or Fahrenheit, required to reduce the decimal reduction by a factor of 10, which is decrease the microbial population by 90%, essentially indicating the organism's thermal resistance. The table below shows a typical example of minutes of exposure and the microbial number at the start of the minute and the microorganisms killed in 1 minute or 90% of the total. And this is the viable microorganisms at the end of 1 minute. And this is the log 10 of the survivor. So when you're calculating the D value, just remember that it is in the 90% of the death, which is one order of magnitude in the logarithmic scale. These complex processes are regulated by two U.S. federal agencies. Environmental Protection Agency EPA regulates disinfectants, and the Food and Drug Administration regulates the agents used on humans and animals. These agencies establish guidelines under which these agents are used and agent effectiveness is measured. The conditions influencing the effectiveness of antimicrobial activity depends on the population size. Larger the population, take longer to kill than smaller populations. Population composition, microorganisms differ markedly in their sensitivity to antimicrobial agents, endospores more resistance, for an example. Concentration or intensity of an antimicrobial agent, usually higher concentration kill more rapidly. These are not linear relationship. Small changes can have large effect. Contact time, the longer the exposure, the more organisms killed. Temperature, higher temperature enhance chemical activity. Local environment, population to be controlled is not isolated, but surrounding by environmental factors that can either prevent or aid destruction. pH, viscosity, and the concentration of organic matter is also important, and organisms in biofilms are less susceptible to many antimicrobial agents. The learning objectives of section 13.2 focus on understanding and comparing different physical methods for controlling microbial growth, which include heating, refrigeration, freezing, high-pressure treatment, desiccation, lyophilization, irradiation, and filtration, etc. For thousands of years, humans have utilized various physical methods for microbial control in food preservation, including high-temperature radiation, filtration, and desiccation. These techniques generally work by non-specifically killing cells through disruption of membranes, altering membrane permeability, or damaging proteins and nucleic acid via processes like denaturation, degradation, or chemical modifications. This section elaborates on the different physical methods employed for effective microbial control. Heating is one of the oldest methods of microbial control, commonly used in cooking and canning. It kills microbes by altering membrane and denaturing proteins, with thermal death point TDP being the lowest temperature that eliminates all microbes in a 10-minute exposure. Microorganisms respond differently to high temperatures, with some like endospore formers, example Clostridium botulinum, being more heat resistant. The thermal death time TDT indicates how long it takes to kill all microbes at a specific temperature. While boiling, a traditional method of moist heat control, effectively kills vegetative cells and some viruses, it is less effective against endospores, which can survive boiling for up to 20 hours. Additionally, boiling efficacy diminishes at higher altitude due to lower boiling points, making it inadequate sterilization technique in laboratory or clinical settings. Heating protocols for sterilization in laboratories and clinics can be categorized into dry heat and moist heat sterilization. Aseptic techniques often utilize dry heat methods, like the direct application of high heat for sterilizing inoculating loops, with incineration effectively eliminating all microorganisms. Dry heat can also be applied for extended periods, typically at temperatures up to 170 degrees Celsius for at least 2 hours using a dry heat sterilizer. The dry heat method can be applied to many liquid media, which can be sterilized in the presence of moist heat. Moist heat sterilization is generally considered more effective due to its superior ability to penetrate cells compared to dry heat when used under pressurized conditions. The slide outlines some of the most heat application temperatures, like boiling for different types of vegetative cells, spores, viruses, and prions. Please note that a moist heat temperature of more than 100 degrees Celsius in the table indicate that a pressurized system must be used, such as an autoclave. Boiling water temperature in an open container can't attain the superheated steam temperature. Autoclaves operate based on the principle of moist heat sterilization, enabling the sterilization of various items, including surgical equipment, by raising temperatures above the boiling point of water. This method effectively eliminates vegetative cells, viruses, and particularly resilient endospores, which can survive standard boiling. The modern autoclave was designed by Charles Chamberlain in 1879 while he was in Louis Pasteur's laboratory and remains the most effective sterilization method available today. In industrial setting, larger autoclaves known as retorts facilitates moist heat sterilization on a grand scale. The functioning of an autoclave involves removing air from its chamber and replacing it with steam, which increases the interior pressure and temperature. There are two primary types of autoclaves: gravity displacement and pre-vacuum sterilizers. Gravity displacement autoclave introduces steam from the top, causing heavier air to sink and exit through a vent at the bottom. However, this method can be less effective for larger loads, necessitating longer sterilization cycles. Conversely, pre-vacuum sterilizer utilize a vacuum to completely remove air before steam introduction, allowing for better penetration of steam into wrapped items. Many autoclaves can operate both methods, with gravity displacement used for decontaminating waste and pre-vacuum for sterilizing prepackaged instruments. Typically, autoclaves operate at standard temperatures of 121 degrees Celsius or 132 degrees Celsius under pressures of 15 to 20 pounds per square inch or psi. The required exposure time for sterilization varies based on the material's volume, nature, and generally lasting at least 20 minutes or more. For effective sterilization, the steam must directly contact the item, which is why the containers are kept loosely closed and instruments loosely wrapped. Achieving the right temperature is crucial as it ensures the destruction of endospores, which is essential for complete sterilization. Quality control is critical in sterilization processes to ensure safety in medical and laboratory settings. Autoclaves are often equipped with recorders to document pressures and temperature during each cycle. Additionally, internal indicators are used to verify that proper sterilization temperature have been met. Common indicators include heat-sensitive autoclave tape, which changes color when a correct temperature is achieved, that does not include exposure duration. Biological indicators such as spore test using Geobacillus styrothermophilus are considered the gold standard for verifying sterilization effectiveness. Geobacillus is a non-pathogenic thermophile, and it will grow within a temperature range of 30 to 75 degrees Celsius. The biological indicator contains spores of organisms on filter paper inside a vial. These tests involve incubating the spores after autoclave cycle to check for bacterial growth, indicating whether the sterilization process was successful or not. Another indicator, the DIAC tube, contains a temperature-sensitive pellet that melts at the appropriate sterilization temperature, providing further assurance of proper autoclave function. Complete sterilization is ideal for many medical applications, but is not always practical for other uses, particularly in food processing, as it may compromise food quality. Methods like boiling and autoclaving can ruin consistency and sensory qualities of food. Pasteurization, developed by Louis Pasteur in the 1860s, offer a solution by using heat to control microbial growth without rendering food sterile. This process effectively kills pathogens and reduces spoilage causing microbes while preserving the overall quality of food. It is most commonly applied to milk and other heat-sensitive products like apple juice and honey. Despite its effectiveness, pasteurized products are not sterile and will eventually spoil. In milk pasteurization, the balance between temperature and treatment time is crucial. One common method is high temperature short time HTST pasteurization, which heats milk to 70 degrees Celsius for 15 seconds, effectively lowering bacterial numbers while maintaining milk quality. Another method, ultra-high temperature UHT pasteurization, involves heating milk to 138 degrees Celsius for a minimum of 2 seconds, allowing it to be stored long-term in sealed containers without refrigeration. However, this method alters the proteins in the milk, which can affect its taste and smell. UHT pasteurization is particularly beneficial in areas with limited access to refrigeration, making it a practical choice for extending shelf life. High temperatures are known to effectively control microbial growth, but low temperature can also serve as a simple and effective method of microbial control, except for psychrophiles that thrive in cold environments. Refrigerators in homes and laboratories typically maintain temperatures between 0 degrees Celsius and 7 degrees Celsius, which significantly inhibits microbial metabolism. This slowed growth helps in preserving refrigerated products such as food items and medical supplies. Additionally, certain laboratory cultures can be stored in these conditions for future use. Freezing temperatures below -2 degrees Celsius can halt microbial growth and may eliminate susceptible organisms. The U.S. Department of Agriculture specifies that safe methods for thawing frozen food include using a refrigerator, immersing them in cold water while changing the water every 30 minutes, or utilizing a microwave, ensuring that food is kept at temperatures that do not encourage bacterial growth. It is important to note that bacterial growth can resume in thawed food, necessitating that these items be treated as fresh perishables. For long-term storage or transport, bacterial cultures and medical specimens are often preserved at ultra-low temperatures of -70 degrees Celsius or lower. Such low temperatures can be achieved by using dry ice in ultra-low freezers or through special liquid nitrogen tanks that maintain temperatures below -196 degrees Celsius, as illustrated in the accompanying slide figure. This method ensures the viability and safety of sensitive biological materials through storage and transport. High-pressure exposure is effective in killing many microbes, making it a valuable tool in the food industry. High-pressure processing, or HPP, is also known as pascalization, is employed to eliminate harmful bacteria, yeast, molds, and parasites and viruses from food products while preserving their quality and extending their shelf life. The technique involves applying pressure between 100 to 800 megapascal, which is significantly higher than the sea level atmospheric pressure of about 0.1 megapascal. This level of pressure is effective in destroying vegetative cells by causing protein denaturation. However, it may not be sufficient to eliminate endospores. While high-pressure processing is advantageous for food safety, it is not typically used for disinfecting or sterilizing surfaces or fomites. In contrast, autoclaving, which combines pressures and steam, is an effective method for killing endospores. In this process, it is primarily the high temperature achieved that led to death to the endospores rather than the pressure itself. Hyperbaric oxygen therapy is utilized in clinical settings to treat infections by allowing patients to breathe pure oxygen at pressures between 1 and 3 atmospheres, typically in a hyperbaric chamber or through a breathing tube. This therapy increases oxygen saturation in hypoxic tissues due to infection and inflammation, enhancing the immune response. through heightened activity of neutrophils and macrophages, which are crucial white blood cells in fighting infections. Additionally, elevated oxygen levels promote the formation of toxic free radicals that inhibit the growth of anaerobic bacteria such as Clostridium perfringens, which is linked to gas gangrene. In cases of Clostridium perfringens infection, this therapy can also diminish the secretion of a bacterial toxin responsible for tissue damage and improve the effectiveness of antibiotics. However, there are rare risks associated with hyperbaric oxygen therapy, including oxygen toxicity and potential harm to sensitive tissues like eyes, middle ear, and lungs due to increased air pressure. Here in this picture, a topical application of the oxygen therapy is shown in a patient with gas gangrene. Drying, also referred to as desiccation or dehydration, is an ancient method used to preserve various foods, including raisins, prunes, and jerky. This process is effective because all cells, including microbes, require water for metabolism and survival. While drying helps control microbial growth, it does not necessarily eliminate all microbes or their endospores, which can revive when conditions become more favorable and water is reintroduced. There are different methods of drying food, such as sun drying or oven drying, which relies on evaporation. The water content of food can be reduced without physical drying by adding solutes like salts or sugars, a process that lowers water activity. High concentration of these solutes create an environment that draws water out of microbial cells, significantly reducing their survival chances. For instance, honey, which consists of about 80% sucrose, inhibits most microorganisms, eliminating refrigeration needs. Salted meats and fish were vital preservation methods before refrigeration, while sugar was used to make jams and jellies. However, some microorganisms, particularly molds and yeasts, can tolerate desiccation and high osmotic pressure, posing a risk of contamination in preserved foods. In addition to conventional drying methods, industries and laboratories use a technique known as freeze drying or lyophilization. In freeze drying, items are quickly frozen and placed under a vacuum to remove water through sublimation. This method effectively combines cold exposure and desiccation, causing less damage to the food compared to traditional methods and better retaining its original qualities. Properly packaged lyophilized items can be stored at room temperature, making this technique popular in both the food industry and the laboratories for long-term storage of microbial cultures. Most of the wet food in space that astronauts eat is freeze-dried. They have to be reconstituted with hot water prior to consumption. In the slide figure A, the working principle of a lyophilizer is shown. First, the substance to be dried is quickly frozen by exposure to liquid ethane or dry ice. Then it is attached to the lyophilizer, shown in figure A as orange-gray round flask, or can be placed in the chamber as shown in the figure C, and frozen below its freezing point, solidifying its water content. Next, a vacuum is applied, reducing moisture and creating conditions for sublimation, where frozen water transitions directly to vapor without becoming liquid. This vapor is removed, leaving freeze-dried product. After most water has sublimed, residual moisture is eliminated through desorption, where the temperature is gradually raised to encouraging remaining water molecules to escape. Finally, the temperature and the pressure are adjusted to ensure complete moisture removal, enhancing the product's shelf life and stability. Radiation in its various forms can effectively kill microbes or inhibit their growth. Ionizing radiation, which includes X-rays, gamma rays, and high-energy electron beams, is particularly powerful as it can penetrate cells and alter their molecular structure. This type of radiation can cause significant damage to cellular components, introducing double-strand breaks in DNA molecules. Such as breaks may lead to direct mutations, or mutations can occur when the cell attempts to repair the damaged DNA. Over time, accumulation of these mutations can result in cell death. In laboratory settings, ionizing radiation is frequently employed to sterilize materials that cannot withstand autoclaving. such as plastic petri dishes and disposable inoculating loops. Clinically, it is used to sterilize various latex and plastic items, including gloves and intravenous tubing, which are essential for patient care. Additionally, ionizing radiation plays a crucial role in sterilizing delicate, heat-sensitive material such as tissues for transplantation, pharmaceutical products, medical equipment, ensuring that these items are safe for use. In Europe, gamma radiation is commonly utilized for food preservation, although its adoption in the United States has been slower. Packaged dry spices are often subjected to gamma irradiation as well. Due to their ability to penetrate various materials, including paper, plastic, and thin metals, strict precautions must be taken when using X-ray and gamma irradiation. These forms of ionizing radiations are unable to penetrate thick layers of iron or lead, which are typically employed as protective barriers for individuals who might be exposed to radiations. UV light is a non-ionizing radiation and an effective tool for controlling microbial growth, utilized by both consumers and laboratory personnel. It is now commonly found in water purification systems for homes, as well as in a portable UV light that campers use to purify water from natural resources. Additionally, germicidal lamps which emit UV at a wavelength of 260 nanometer are employed in surgical suites, biological safety cabinets, and transfer hoods. Since UV light does not penetrate surfaces and cannot pass through plastics or glass, it is essential for cells to be directly exposed to the light source for effective microbial control. Sunlight encompasses a broad spectrum including both UV and visible light, which can be effective against certain bacteria. This effectiveness is attributed to the formation of thymine dimers caused by UV light and the production of reactive oxygen products that occur in small amounts when exposed to visible light. Together, these factors contribute to antimicrobial properties of sunlight, enhancing its ability to combat specific microbial threats. Sonication is the process of using high-frequency ultrasound waves to disrupt cell structure by causing rapid pressure changes in intracellular liquid, leading to cavitation and the formation of bubbles that can ultimately cause the cell to lyse or collapse. This technique is valuable in laboratory setting for efficiently lysing cells to release their content for research. Additionally, sonication is employed outside the lab for cleaning surgical instruments, lenses, and various objects, including coins, tools, and musical instruments. Filtration is a method of physically separating microbes from samples. Air is commonly filtered through high-efficiency particulate air or HEPA filters, as shown in the slide figure. HEPA filters have effective pore sizes of 0.3 micrometer, small enough to capture bacterial cells, endospores, and many viruses, as air passes through these filters, nearly sterilizing the air on the other side of the filter. HEPA filters have a variety of applications and are used widely in clinical settings, in cars, airplanes, and even in the home. For example, they may be found in vacuum cleaners, heating and air conditioning systems, and air purifiers. HEPA filters are commonly used in hospitals and surgical suites to prevent contamination and spread of airborne microbes through ventilation systems. HEPA filtration systems may be designed for entire buildings or individual rooms. For example, burn units, operating rooms, or isolation units may require special HEPA filtration systems to remove opportunistic pathogen from the environment because patients in these rooms are particularly vulnerable to infection. Biological safety cabinets effectively utilize HEPA filters to ensure a clean and safe working environment when handling biological materials. These filters serve to remove particulates from air entering or exiting the cabinet. An air intake HEPA filter is crucial for preventing environmental contaminants from entering the biological safety cabinets, while an air exhaust HEPA filter protects the laboratory by preventing pathogens from escaping. This dual function helps maintain a safe workspace for laboratory personnel. There are three classes of biological safety cabinet: Class 1, 2, and 3, each offering varying levels of protection for laboratory workers and the environment. Class 1 biological safety cabinets provide a low to moderate level of protection against biological agents, with air being drawn in filtered, and then exhausted through the building's system. Class 2 biological safety cabinet enhance safety with directional airflow and partial barriers to contain infectious agents. Class 3 biological safety cabinets are specifically designed for handling highly infectious agents found in BSL-4 laboratories, offering the highest level of protection. Class 3 biological safety cabinets are gas-tight and required materials to pass through a double-door system for decontamination before entering or exiting the cabinet. Air is filtered through 1 or 2 HEPA filters and subjected to an incineration system before being released outdoors. Personnel working within Class 3 cabinets can manipulate materials using long rubber gloves that are sealed to the cabinet, ensuring maximum containment and safety while handling hazardous biological materials. Filtration can effectively remove microbes from liquid samples through membrane filtration, which operates similarly to HEPA filters for air. Membrane filters designed to eliminate bacteria generally have an effective pore size of 0.2 micrometer, smaller than the average bacterium size of about 1 micrometer. Although smaller pore sizes are available for specialized applications. This method is particularly beneficial for filtering bacteria from heat-sensitive laboratory solutions such as antibiotic and vitamin solutions, and large volumes of cell culture media can be filter sterilized instead of autoclaved to safeguard these components. While syringe filters are commonly used for small volumes, vacuum filters are preferred for large volumes, as shown in the figure A. The table provides a comprehensive summary of physical methods of microbial control, the different conditions, mode of actions, and examples. The table of the physical methods of microbial control is continued in this slide too. The learning objectives focus on understanding the comparing different chemicals used for controlling microbial growth. This includes examining their uses, advantages, disadvantages, chemical structures, and modes of action. Chemical methods are utilized along with physical methods for microbial control, employing a diverse range of disinfectants and antiseptics. Selecting the appropriate chemical involves considering the targeted microbe, the required cleanliness of the item, and the potential impact on the item's integrity, safety for humans, animals, and the environment, cost, and usability. This section outlines various chemicals used as disinfectants and antiseptics, detailing their mechanism of action and typical applications. In the 1800s, scientists began exploring various chemicals for disinfection purposes. British surgeon Joseph Lister played a crucial role in this development during the 1860s by utilizing carbolic acid or phenol as the disinfectant for surgical wounds. His work significantly influenced American chemist Joseph Lawrence, who in 1879 created Listerine, an alcohol-based antiseptic still in use today for oral hygiene. While carbolic acid has fallen out of favor as a surgical disinfectant due to its skin irritant properties, the chemical compounds found in modern antiseptic mouthwashes and throat lozenges are classified as phenolics. Phenol, a compound consisting of benzene ring with an OH group, forms the basis for phenolics, which are compounds that include this structure. Naturally occurring phenolics such as thymol and eucalyptol can be found in various plants, while others are derived from creosote, a coal tar component. Phenolics have favored for their chemical stability, persistence on surfaces, and lower toxicity compared to phenol. These compounds combat microbial growth by denaturing proteins and disrupting cellular membranes. Since Lister's era, numerous phenolic compounds have been employed in controlling microbial growth. For instance, cresols and O-phenylphenol have been active ingredients in Lysol formulations since its inception in 1889. O-phenylphenol also found agricultural use in managing bacterial and fungal growth on harvested crops, particularly citrus fruits, though its application in the US is now more restricted. Another notable phenolic compound in hexachlorophene, an active ingredient in pH isohex, a topical detergent widely used in hospitals for hand washing. pH isohex is effective against gram-positive bacteria, including those that cause staphylococcal and streptococcal skin infections. But its use for bathing infants has ceased due to potential neurological risks associated with hexachlorophene exposure. Triclosan, another bisphenol compound, has become widely used in antibacterial products over recent decades. Initially incorporated into toothpaste, it is now prevalent in hand soaps and is often added to various items such as cutting boards, shower curtains, and clothing to impart antimicrobial properties. Triclosan is particularly efficient against gram-positive bacteria on the skin, as well as certain gram-negative bacteria and yeasts, highlighting the broad application in maintaining its hygiene and preventing infections. Some of the first chemical disinfectants and antiseptics used were heavy metals, which kill microbes by binding to the proteins and inhibiting enzymatic activity. These metals are oligodynamic, meaning they even in very small concentration can exhibit significant antimicrobial properties. Heavy metal ions strongly bind to sulfur-containing amino acids and can bioaccumulate within cells, leading to localized concentration that cause proteins to denature. However, heavy metals are not selectively toxic to microbial cells. They can also bioaccumulate in humans and animal cells. Excessive concentration may have toxic effects on humans, such as a silver accumulation leading to erigeria, a condition that causes the skin to turn irreversibly blue-gray. To mitigate the potential toxicity of heavy metals, it's important to carefully control both the duration and exposure and the concentration of heavy metal. Mercury is a heavy metal that is historically been used for controlling microbial growth, notably in the treatment of syphilis for many centuries. Compounds like mercuric chloride exhibit bacteriostatic properties and broad spectrum of antimicrobial activity by binding to sulfur-containing amino acids and proteins, thereby inhibiting their functions. However, the use of mercury compounds has significantly decreased in recent decades due to their toxicity, which poses serious risk to central nervous, digestive, and renal system at high concentrations. The environmental impact of mercury is also concerning, particularly its tendency to bioaccumulate in fish, leading to further health risks. While topical antiseptics such as mercurochrome and merthiolate, which contain mercury in low concentrations, were once commonly used, growing concerns over mercury exposure have led to these products being withdrawn from the market in the United States. Silver has long history as an antiseptic, with ancient practices including storing drinking water in silver jugs. Today, Silvadene cream is frequently used for treating topical wounds, especially burn injuries, to prevent infection. In the past, silver nitrate drops were applied to newborn eyes to prevent ophthalmia neonatorum, although antibiotic creams are now more commonly used. Additionally, silver is often combined with antibiotics to significantly enhance their effectiveness. It is also incorporated into medical devices like catheters and bandages to prevent antimicrobial properties. However, there are concerns that heavy metals like silver could contribute to the development of antibiotic resistance. Several heavy metals such as copper, nickel, and zinc are recognized for their antimicrobial properties. Copper sulfate is frequently employed as an algicide in swimming pools and fish tanks, while metallic copper is increasingly used in reduced microbial growth. For instance, copper lining in the incubators help minimize contamination in cell cultures, and the use of copper pots for water storage in underdeveloped regions is being explored as a means to combat diarrheal diseases. Additionally, copper coatings are gaining popularity for high-touch surfaces like doorknobs and cabinet hardware in healthcare settings to limit the spread of microbes. Nickel and zinc coatings are also being applied for similar antimicrobial purposes. Various forms of zinc, including zinc chloride and zinc oxide, are widely used in commercial products. Zinc chloride is known for its safety and commonly included in the mouthwashes, enhancing their effectiveness. Meanwhile, zinc oxide is found in numerous items such as topical antiseptic creams, calamine lotion, diaper ointments, baby powder, and dandruff shampoos, showcasing the versatility and safety of zinc in promoting health and hygiene. Disinfection is often achieved using halogens such as iodine, chlorine, and fluorine. Iodine acts as an oxidizing cellular components like sulfur-containing amino acids, nucleotides, and fatty acids, which destabilizes micromolecules. While iodine is frequently utilized as a topical tincture, it can cause staining and skin irritation. To improve its stability and efficacy, iodine is often complexed with organic molecules forming iodophores such as povidone iodine. A well-known example is betadine, commonly employed by medical personnel for hand scrubs before surgeries and for antiseptic preparations on patients' skin. Chlorine, another prevalent halogen, produces hypochlorous acid when mixed with water, which is an effective antimicrobial agent. The chemical is widely used in municipal drinking water system and wastewater treatment facilities, necessitating precautions to minimize worker exposure to chlorine gas. Sodium hypochlorite, the active component in household bleach, serves various disinfection purposes, including swimming pool sanitation. Additionally, chlorine-based disinfectants are essential in the food processing and restaurant industries to combat foodborne illnesses. A statement from the Food and Agricultural Organization (FAO) and the World Health Organization reassured that the safe use of chlorine products in food processing does not pose risk to consumers. Chloramines are a class of chlorinated compounds utilized primarily as disinfectants. They are stable and release chlorine over extended periods, making them effective in this role. Derived from ammonia, chloramines are formed when 1, 2, or all 3 hydrogen atoms in ammonia are replaced with chlorine atoms. Their stability and disinfecting properties make them suitable to various applications, particularly in water treatment. These compounds, along with other chlorine derivatives, are used for disinfecting drinking water. Chloramine tablets are especially common in military settings for these purposes. In emergencies such as natural disaster that compromises public water supplies, the CDC recommends disinfecting tap water with small amounts of household bleach. Recent studies highlight sodium dichloroisocyanurate, or NADCC, as a viable alternative for drinking water disinfections. NADCC tablets are now available for civilian and military use, and they are considered preferable to chloramine tablets for emergency situations. Additionally, chlorine dioxide is frequently utilized for water disinfections and for fumigation in enclosed areas. Despite their effectiveness, chlorinated compounds come with certain drawbacks. Some individuals may experience irritation to skin, nose, or eyes when exposed to these disinfectants. Furthermore, chlorinated compounds may not eliminate all pathogens, particularly resilient organisms like protozoan parasite Cryptosporidium, which has a protective shell that resists chlorine. In emergency scenarios, boiling water is recommended as a more effective method of disinfection when possible. Fluorine, another halogen, is recognized for its antimicrobial properties and its role in preventing dental caries. Fluoride, the active ingredient in toothpaste, is often added to public water supplies to support oral health. It integrates into the hydroxyapatite structure of tooth enamel, enhancing its resistance to acid attacks from oral bacteria. Moreover, fluoride promotes the uptake of calcium and phosphate ions, aiding in the remineralization of enamel. Beyond strengthening teeth, fluoride also exhibits bacteriostatic effects by accumulation in plaque-forming bacteria, disrupting their metabolism, and thereby reducing acid production linked to tooth decay. Alcohols are a significant group of chemicals utilized as disinfectants and antiseptics. They function primarily by rapidly denaturing proteins, which inhibit cellular metabolism and disrupting cellular membranes, leading to cell lysis. Alcohols are most effective in approximately 70% aqueous solutions, as higher concentration can cause rapid coagulation of surface proteins, hindering adequate penetration into cells. Ethyl alcohol or ethanol and isopropyl alcohol or isopropanol are the most commonly used alcohols in disinfection processes. These alcohols are generally bactericidal and fungicidal with some effectiveness against enveloped viruses, though they are not sporicidal. They can inhibit sporulation and germination processes. Despite the fast evaporation and drying properties, alcohol may irritate the skin due to dehydration at application site. A typical clinical application includes swabbing the skin to prepare for needle injections, and they are also key ingredients in popular instant hand sanitizer. However, these sanitizers are less effective when visible dirt is present. Additionally, alcohols are used to create tinctures with other antiseptics such as iodine tinctures. They are relatively inexpensive and effective against a wide range of vegetative microbes. However, their high volatility poses limitations, reducing their effectiveness to shortly after application. Surface active agents or surfactants are chemical compounds that reduce the surface tension of water and are key components in soaps and detergents. Soaps, which are salts of long-chain fatty acids, possesses both polar and nonpolar regions that enable to interact with a variety of substances, including nonpolar oils and grease. This interaction facilitates the creation of emulsions in water, helping to loosen and remove dirt and microbes from surfaces and skin. Although soaps do not kill microorganisms, their mechanical action allows for effective degerming. Some soaps include added bacteriostatic agents like trichlorocarbon or chlorfluorocarbon, which provides additional antiseptic or disinfectant properties. While soaps are effective, they often leave behind films that are tough to rinse away, especially in hard water, which contains high level of calcium and magnesium salts. In contrast, detergents are made from synthetic surfactants that maintain strong cleansing capabilities, are more soluble, thereby preventing soapy residues. Anionic detergents commonly used for laundry feature a negatively charged anion linked to a long hydrophobic chain, whereas cationic detergents have positively charged cation. This difference in charge significantly affects their properties and applications. Cationic detergents include quaternary ammonium salts, quarts, which serves as important disinfectants and antiseptics. Quarts have a quaternary nitrogen atom that gives a positive charge, allowing them to disrupt bacterial phospholipid bilayers and compromise membrane integrity. These compounds are stable, non-toxic, and effective against a range of microorganisms, including bacteria, fungi, protozoans, and enveloped viruses, although they do not affect endospores. Quarts are commonly found in clinical settings and household cleaners, such as Lysol products, where benzalkonium chlorides acts as the active ingredient in skin antiseptics and mouthwashes. Bisbiguanides, first synthesized in the 20th century, are cationic molecules with notable antiseptic properties, notably chlorhexidine. This antiseptic exhibits broad-spectrum activity against various microorganisms, including yeasts and both gram-positive and gram-negative bacteria, although it is ineffective against Pseudomonas aeruginosa due to the potential resistance. Chlorhexidine disrupts cell membranes, act as a bacteriostatic at low concentration and bactericidal at high concentrations, and it is also effective against enveloped viruses. However, it is less effective against Mycobacterium tuberculosis and non-enveloped viruses and does not kill spores. Commonly used in clinical settings for surgical scrubs and hand washing, chlorhexidine offers long-lasting antimicrobial activity and can be used as an oral rinse. Another bisbiguanide, alexidine, is emerging as a faster-acting alternative for surgical scrubs and oral rinses. Alkylating agents are potent disinfectants and functions by substituting a hydrogen atom in a molecule with an alkyl group, leading to the inactivation of enzymes and nucleic acids. One of the most notable alkylating agent is formaldehyde, which is typically utilized in a 37% solution known as formalin or as a gaseous disinfectant. It offers a broad-spectrum efficacy against bacteria, viruses, fungi, and endospores, allowing for sterilization at low temperatures, a beneficial alternative to more labor-intensive heat sterilization methods. Formaldehyde is also employed as a chemical fixative for tissue specimen storage and in vaccine preparation. However, due to its irritating nature and carcinogenic properties, it is not suitable for use as an antiseptic. Another important alkylating agent is glutaraldehyde, which is similar in structure to formaldehyde but features two reactive aldehyde groups, enabling it to act more swiftly. Commonly available in a 2% solution marked as Cidex, it is also primarily used for disinfecting surfaces and sterilizing medical equipments. Despite its effectiveness, glutaraldehyde also causes skin irritation and is not recommended for antiseptic purposes. A newer disinfectant, othalaldehyde (OPA), has emerged as a replacement for glutaraldehyde in disinfecting medical equipment. Like glutaraldehyde, OPA contains two reactive aldehyde groups, but they are connected by an aromatic bridge. The OPA is believed to operate similarly to both glutaraldehyde and formaldehyde while offering advantages such as reduced skin irritation, minimal odor, and necessity for pre-processing. Additionally, OPA is more effective against mycobacteria, making it a favorable choice for medical settings. Lastly, beta-propionlactone is an alkylating agent that differs chemically from the aforementioned agents. It functions by binding to DNA to inactivate it and capable of eliminating endospores. This clear liquid with a strong odor is utilized in both liquid and vapor forms for sterilization of medical instruments, tissue grafts, and various biological materials. Although it is rapidly metabolized to lactic acid in humans and animals, beta-propionlactone is also an irritant and poses significant health risks, including potential permanent damage to organs and carcinogenicity in animals, necessitating precautions to limit human exposure. Sterilizing gases can be used to sterilize heat-sensitive materials. These gases are microbicidal and sporicidal. Ethylene oxide represents another category of alkylating agents used for gaseous sterilization. Its high penetrating capability allows for sterilization of heat-sensitive items, including plastic-wrapped medical devices and laboratory supplies. Ethylene oxide is recognized as a form of cold sterilization. However, it is also carcinogenic and highly explosive. When properly handled and aerated, post-treatment ethylene oxide proves to be an effective sterilization method commonly used in medical environments. In addition, chlorine dioxide, vaporized hydrogen peroxide are also used for their sterilizing heat-sensitive materials. Peroxygens are powerful oxidizing agents commonly utilized as disinfectants and antiseptics, with hydrogen peroxide being the most prevalent. This compound is often employed in solutions form to disinfect surfaces and can also be used in gaseous states. Hydrogen peroxide is a cost-effective skin antiseptic that decomposes into water and oxygen, making it environmentally friendly. However, its decomposition is hastened by light, leading to the necessity of storing it in a brown or opaque bottles. Despite its benefit, hydrogen peroxide can harm the skin, potentially delaying healing and cause scarring, which is a significant drawback. Additionally, it is frequently included in contact lens cleaner and disinfectant properties. The mechanism of action for hydrogen peroxide involves the generation of free radicals that damage cellular macromolecules. It exhibits broad-spectrum antimicrobial activity, effectively targeting gram-positive and gram-negative bacteria, fungi, viruses, and endospores, with slightly higher effectiveness against gram-positive bacteria. However, certain bacteria that produce the enzymes catalase or peroxidase can tolerate low concentrations of hydrogen peroxide. To effectively kill endospores, an increase in either the duration of exposure or the concentration of the hydrogen peroxide is required. Gaseous hydrogen peroxide is particularly potent and can serve as a sterilizing agent for rooms or medical equipment. Plasma, recognized as the fourth state of matter, is another effective sterilization method due to its ability to penetrate surfaces and eliminate both vegetative cells and endospores. Both hydrogen peroxide and peracetic acid can be introduced as plasma for sterilization purposes. Peracetic acid in particular is noteworthy because it can be used in either liquid or plasma form, demonstrating superior efficacy over hydrogen peroxide even at low concentrations. Additionally, peracetic acid remains unaffected by catalases and peroxidases, allowing it to effectively kill endospores. Other peroxygens include benzoyl peroxide and carbamide peroxide, which serve specialized roles. Benzoyl peroxide is found in acne treatment, targeting the bacterium Propionibacterium acnes, while carbamide peroxazole is an ingredient in a toothpaste that helps combat oral biofilms linked to tooth discoloration and bad breath. Ozone gas, another peroxygen, is utilized for its disinfectant properties to purify air and water supplies. Overall, peroxygens are recognized for their high effectiveness and widespread application, boasting minimal environmental hazards. The use of supercritical fluids, particularly supercritical carbon dioxide, has become increasingly popular sterilization purposes over the past few decades. When carbon dioxide is subjected to pressure around 10 times that of the atmosphere, it transitions into a supercritical state, exhibiting properties that are a blend of both liquid and gases. This unique behavior allows the supercritical carbon dioxide to effectively penetrate surfaces, making it an effective sterilizing agent for various materials placed in pressurized chambers. The mechanism by which supercritical carbon dioxide sterilizes involves its ability to infiltrate cells, generate carbonic acid, significantly lowering the pH within the cells. This process is particularly effective against vegetative cells and can be enhanced by combining supercritical CO2 with peracetic acid to target resistant endospores. Additionally, increasing the temperature or employing rapid cycles of pressurization and depressurization can further improve its efficacy by promoting cell lysis. One of the key advantages of the using supercritical CO2 for sterilization is its non-reactive, non-toxic, and non-flammable nature, allowing for effective sterilization at lower temperatures. Unlike traditional methods such as heat or irradiation that may compromise the integrity of sterilized objects, supercritical carbon dioxide preserves the quality of materials, making it suitable for food treatment and medical devices like endoscopes. Moreover, it is increasingly being utilized for disinfecting tissues prior to transplantation and for pest control, and it can eliminate insect eggs and larvae in various products. Chemical preservatives play a crucial role in food preservation by inhibiting microbial growth and minimizing spoilage. Common preservatives such as sorbic acid, benzoic acid, and propionic acids are frequently used in various foods, particularly those that are acidic. Their more soluble salts, including potassium sorbate, sodium benzoate, calcium propionate, are effective in controlling the growth of molds. Importantly, these substances are non-toxic, easily metabolized by humans, and flavorless, ensuring that they do not alter the taste of the preserved foods. Sorbic acid and benzoic acid demonstrate enhanced effectiveness at lower pH levels. Sorbic acid is known to inhibit several cellular enzymes, including those involved in the citric acid cycle, as well as catalases and peroxidases. It is commonly used in a range of foods such as dairy products, bread, fruits, and vegetables. Conversely, benzoic acid occurs naturally in many fruits and berries, as well as in spices and fermented products. It is believed to lower intracellular pH, disrupting essential cellular processes like oxidative phosphorylation and amino acid uptake. Foods that utilize benzoic acid or sodium benzoate for preservation, including fruit juices, jams, and ice creams, pastries, soft drinks, chewing gum, and pickles. Propionic acid functions similar to benzoic acid by inhibiting enzymes and lowering intracellular pH, although it is more effective as a preservative at higher pH levels. This acid is naturally produced during the ripening of certain cheeses and is added to the various cheeses types and baked goods to prevent mold growth. Additionally, propionic acid is used in raw dough to combat the bacterium Bacillus mesentericus, which can spoil bread by making it ropey. Other chemical preservatives like sulfur dioxide and nitrites are also widely used. Sulfur dioxide prevents browning in foods and is particularly effective in preserving dried fruits, with a historical use in winemaking. While sulfites can be metabolized by the human body, they can cause allergic reaction in some individuals, particularly those with asthma, and can degrade thiamine, an essential nutrient. The exact mechanism of action for sulfites remains somewhat unclear, although they may interfere with protein bond formation and cellular processes by reducing intracellular pH. Nitrites, commonly found in processed meats, can maintain the color and inhibit germination of Clostridium botulinum endospores. They are converted to nitric oxide, which interacts with the heme group in the meat, giving it a red color. Additionally, nitric oxide can disrupt essential bacterial enzymes, preventing ATP synthesis. However, nitrosamines, which are carcinogenic compounds, can form when nitrite-preserved meats are exposed to heat during cooking, posing potential health risks associated with consumption of products like hot dogs, lunch meats, breakfast sausages, and bacon. The discovery of natural antimicrobial substances produced by other microbes has added to the arsenal of preservatives used in food. Nicene is an antimicrobial peptide produced by the bacterium Lactococcus lactis and is particularly effective against gram-positive organisms. Nicene worked by disrupting cell wall production, leaving cells more prone to lysis. It is used to preserve cheeses, meats, and beverages. Natamycin is an antifungal macrolide antibiotic produced by bacterium Streptomyces natalensis. It was approved by the FDA in 1982 and used to prevent fungal growth in various types of dairy products, including cottage cheese, sliced cheese, and shredded cheese. Natamycin is also used for meat preservation in countries outside the United States. This slide provides a comprehensive summary of all the chemical disinfectants that we have discussed so far. This comprehensive summary of the chemical disinfectants continued, and this slide concludes the comprehensive summary of the chemical disinfectants. The learning objective of this section will include understanding of the phenol coefficient, disk diffusion, dilution, and in-use methods for antiseptic disinfectants and sterilants. The phenol coefficient test is a laboratory method designated to assess the antimicrobial activity of disinfectants and antiseptics by comparing their germicidal potency to phenol, which serves as a standard disinfectant. The procedure begins with the preparation of serial dilutions of the test substance and phenol in distilled water, typically ranging from 1:100 to 1:1000. A standardized suspension of bacteria, often Salmonella typhi or Staphylococcus aureus, is then inoculated into each dilution. The tubes are incubated at 37 degrees Celsius for varying durations of 5, 10, 15, and 20 minutes, after which they are examined for bacterial growth. The highest dilution that effectively kills the bacteria within the specified time is noted for both the test substance and the phenol, with the phenol coefficient calculated by dividing the dilution of the test substance by the dilution of phenol. A coefficient greater than 1 signifies that the test substance is more potent than phenol, while a coefficient of 1 indicates equal potency, and less than 1 shows reduced potency. However, the test has limitation, as it relies on phenol, which may not represent all disinfectants, and the results may not accurately reflect real-world efficacy due to laboratory conditions, excluding factors like sporicidal or virulence activity and stability. In conclusion, while phenol coefficient tests offers traditional measures of antimicrobial potency relative to phenol, it should be interpreted cautiously due to its inherent limitations. The disk diffusion method involves applying various chemicals to sterile filter paper discs, which are then placed on an agar plate inoculated with a target bacterium. As the bacteria grow, the chemicals diffused into the agar creating a clear zone of inhibition around the discs where microbial growth is suppressed. The size of these zones, influenced by factors like chemical solubility and diffusion ability, typically indicates the effectiveness of a chemical agent, with larger zones suggesting greater inhibitions. The diameter of each zone is measured in millimeters. The effectiveness of chemical agents in clinical settings is often assessed using various methods, including the use dilution test. The test evaluates a chemical's disinfection capability on inanimate surfaces by dipping a stainless steel cylinder into a culture of target microorganisms, allowing it to dry, and then immersing it in disinfected solutions at different concentrations for a set period. Afterward, the cylinder is placed into a sterile medium devoid of disinfectants and incubated. If bacterial survival occurs, it is indicated by turbidity in the medium, while ineffective disinfection is shown by absence of turbidity. To ensure reliability, the Association of Official Agricultural Chemists International (AOAC) has established that at least 59 out of 60 replicates must show no growth for a passing result. Additionally, the results should be consistent across different disinfectant batches and testing days. Manufacturers utilized these use dilutions to substantiate the efficacy claim of their products as required by the EPA. An in-use test assesses the microbial contamination of disinfectants actively used in clinical settings. In these procedures, a 1 ml sample of disinfectant is diluted in 9 ml of sterile broth containing a compound to neutralize the disinfectant. Approximately 0.2 ml of this mixture is inoculated onto agar plates, one incubated at 37 degrees Celsius for 3 days and the other at room temperature for 7 days. The growth of 5 or more colonies on either plate indicate the presence of viable microbes, suggesting the disinfectant solution is contaminated. These tests are crucial for monitoring disinfectant effectiveness in clinical environments. Eyes on ethics, contaminated endoscope. Carbapenem-resistant Enterobacteriaceae infections linked to contaminated endoscopes have emerged as a significant issue. Notably, an outbreak at Ronald Reagan UCLA Medical Center in 2015 exposed 179 patients, resulting in infections for 7 and 2 fatalities. Lawsuits against Olympus, the endoscope manufacturer, allege failure to secure FDA approval for design changes and withholding defect information. These lawsuits highlight complex liability questions. While invasive procedures carry risks, strict protocol adherence can minimize negative outcomes. The responsibility for such outcome is debated. Should hospitals or healthcare workers be liable for the following flawed procedures? Should manufacturers be held accountable for defective equipment? The government's role in ensuring the safety of medical protocols and equipment is also scrutinized. The protocol for medical equipment cleaning are often developed by agencies like FDA and organizations like AOAC. End users, primarily hospitals, are responsible for adhering to these protocols and can be liable for breaches. However, protocols can have flaws, as evidenced by inadequate FDA approval disinfection method for endoscope that failed to prevent subsequent CRE outbreaks. In response to these outbreaks, stakeholders are exploring solutions. Hospitals are enhancing cleaning procedures, manufacturers are redesigning duodenoscope to eliminate hard-to-clean areas, and the FDA is revisiting its guidelines. Despite these efforts, there is no guarantee that a new protocol will entirely eliminate infection risks. Endoscopes are used in 500,000 to 650,000 life-saving procedures annually in US, raising the question of when risks may outweigh benefit and who should be responsible for negative outcomes. Clinical focus on nosocomial infection by a carbapenem-resistant enterococci or CRE. Robert is a 46-year-old real estate agent who recently had a cholecystectomy a surgery to remove painful gallstones. The procedure was performed laparoscopically with the assistance of a duodenoscope, a specialized endoscope that allows surgeons to visualize the interior of the body through a tiny camera. After returning home from the hospital, Roberta experienced a significant abdominal pain and high fever, along with burning sensation during urination and blood in her urine. Following her post-operative instructions, she promptly informed her surgeons of these concerning symptoms. Upon evaluation, Roberta's physician suspected a bacterial infection due to the sudden onset of her high fever, abdominal pain, and bloody urine, leading to a diagnosis of urinary tract infections. UTIs can be caused by various bacteria, typically when bacteria from lower gastrointestinal tract enter the urinary tract. However, given Roberta's recent surgery, the physician was concerned that she may have contracted a nosocomial or hospital-acquired infection. A urine sample was collected and the urine culture was ordered to identify the presence of white blood cells, red blood cells, and bacteria, while a course of antibiotic ciprofloxacin was prescribed to address the infection. Despite the antibiotic treatment, Roberta's symptoms continued to worsen, and she subsequently developed pyelonephritis, a severe kidney infection, which necessitated her rehospitalization in the intensive care unit. Pyelonephritis is an inflammation of the kidneys caused by bacterial infection. It is a type of urinary infection that affects the upper urinary tract, which includes the kidneys, ureters, and renal pelvis. Her conditions deteriorated further, leading to symptoms of septic shock. In response, her physicians ordered a urine, culture, and drug sensitivity test to identify the specific cause of her infection and determine effective antibiotics. The results revealed resistance to a wide range of antibiotics, including carbapenems, which are typically reserved for severe infections. These alarming findings indicated that Roberta was infected with a superbug from carbapenem-resistant Enterobacteriaceae family, known for its high resistance to treatment. CRE infections are particularly challenging to manage, with a fatality rate of 40 to 50%. To combat Roberta's kidney infections and septic shock, she underwent treatment that included dialysis, intravenous fluids, and medications to stabilize her blood pressure and prevent clotting. Additionally, she received aggressive treatment with tigecycline, a new drug effective against drug-resistant bacteria. After several weeks in the ICU, Roberta made a recovery from CRE infections, but public health officials soon recognized that her case was a part of a larger issue. Several other patients underwent similar procedures at the same hospital also developed CRE infections, with some fatalities reported. The investigation traced the source of these infections back to the duodenoscope utilized during the surgeries. Although hospital staff adhered to manufacturer's protocol for disinfections, the persistence of CRE bacteria within the instruments highlighted the significant risk in surgical procedure, ultimately affecting multiple patients and raising concerns about the safety of such medical devices. Case in point, botulism outbreak of 2015 in Ohio. Lancaster is a quiet little town of 40,000 southeast of Columbus, Ohio. In spring 2015, after attending a church potluck, a woman experienced blurred vision, difficulty swallowing, and drooping eyelids, prompting her emergency hospitalization. There she developed abdominal cramping, nausea, paralysis, dry mouth, and trouble speaking and breathing. Due to these symptoms, the hospital activated its incident command center, notifying public health officials of a potential botulism case. Other patients with similar symptoms also arrived at local hospitals, leading to overnight shipment of antitoxin from CDC. Tragically, the first patient died from respiratory failure, and about half of others required hospitalization. At least 2 needed ventilators. Public health officials discovered all victims had attended the same church potluck, tracing the outbreak to a potato salad made with home-canned potatoes. The canning method likely used boiling water, which allows Clostridium botulinum endospores to survive. This bacterium produces the deadly neurotoxin. According to the CDC, this incident marked the largest botulism outbreak in the U.S. history in nearly 40 years, affecting 19 individuals. To kill Clostridium botulinum endospores, a minimum temperatures of 116 degrees Celsius is required, achievable only by a pressure canner, recommended for low-acid foods. The CDC also advises boiling home-canned foods for about 10 minutes before consumptions, as the botulinum toxin is heat labile and can be rendered non-functional through heating. Gamma irradiation is a method to prevent food spoilage and foodborne illnesses, yet it faces public skepticism primarily due to misinformation. As an analogy, a patient after an X-ray or after receiving radiation therapy does not become radioactive. This technique involves exposing food to cobalt-60 or cesium-137 in a radiation chamber without direct contact, ensuring that the food does not become radioactive and poses no risk of radioactive exposure when consumed. Nutritional quality remains largely unchanged, with only minor vitamin loss with potential alteration in taste for high-fat foods, which can be minimized by using lower radiation doses at cooler temperatures. In the US, organizations such as CDC, EPA, and FDA endorse the safety and effectiveness of irradiation for various foods, including meats, poultry, seafood, fruits, vegetables, and spices. Many countries, including France, Netherlands, and Australia, have also approved gamma irradiation to enhance consumers' understanding and acceptance. Irradiated foods are labeled with an international symbol, Radura. A recent study suggests a growing acceptance among the consumers. Hand soaps and cleaning products, often labeled as antibacterial, imply superior cleaning compared to conventional soaps. However, the safety and effectiveness of antibacterial ingredients, primarily triclosan, are under scrutiny. Approximately 75% of the antibacterial liquid hand soaps and 30% of the bar soap contain triclosan, which disrupt a specific bacterial enzyme not present in humans. Despite its increasing use since 1990s, over 40 years of FDA research reveals no conclusive evidence that triclosan provides significant health benefit over traditional soap. While some studies show slightly fewer bacteria on hands after using triclosan soap, no reduction in the transmission of illness-causing bacteria has been demonstrated. Concerns regarding triclosan include the emergence of resistant bacterial strain, including Salmonella enterica, which may diminish triclosan's effectiveness over time. Bacteria can develop resistance through minor genetic changes, unlike other disinfectants that require more significant alterations. Additionally, triclosan has accumulated in the environment, disrupting sewage decomposition and harming beneficial bacterial population in water systems. It has been detected in various human fluids, with the CDC study revealing detectable levels in 75% of the tested individuals. In December 2013, the FDA mandated that soap manufacturers demonstrate significant benefits of antibacterial soaps over traditional options by 2016 or face removal from the market. No studies effectively proved the benefit of triclosan in soaps. In September 2016, the FDA banned the use of triclosan in consumer antiseptic wash products like soaps, citing a lack of evidence for their effectiveness and potential safety concerns, including the development of antibiotic resistance. Hand washing is essential for public health, particularly in clinical settings. The CDC advises that the general public wash their hands before, during, after handling food, before eating, before and after interacting with sick individuals, before and after wound treatment, and after using the toilet or changing diapers, after respiratory hygiene, after handling garbage, and after contact with animals or their waste. The CDC outlined 5 key steps for effective hand washing. For healthcare workers, hand washing is crucial between patient interactions, after removing gloves, following contact with bodily fluids, potentially infectious material, and before and after assisting in surgical procedures. Surgical scrubbing is more thorough than regular hand washing, aimed at reducing skin microbiota to prevent infections in surgical wounds. There is no universally accepted surgical scrubbing protocol. The duration and methods can vary based on the antimicrobial agents used. Healthcare workers should adhere to manufacturers' guidelines. According to the Association of Surgical Technologies, Surgical scrubs can be performed with or without brushes. Prions are acellular misfolded proteins that cause incurable fatal disease such as Kuru and Creutzfeldt-Jakob disease. They are extremely resistant to destruction, making them notoriously difficult to eliminate. Prions are not only highly infectious, but also deadly, necessitating stringent handling and disposal procedure for any prion-infected materials. Individuals who work with such materials require extensive training and must exercise extreme caution to avoid potential exposure. Typical disinfection methods include autoclaving and the use of common chemicals like phenol, alcohol, formalin, and beta-propyolactone can reduce but not fully eliminate prion infectivity. Even brain and spinal cord tissues that have been fixed in formalin remain infectious, highlighting the challenges in effectively managing prion-contaminated items. This resilience underscores the need of specialized protocols when dealing with prion contamination. To ensure safety, personnel handling contaminated specimens or equipments must wear protective gear, including coat, face protection, cut-resistant gloves, and in the event of skin contact with prion-contaminated materials, immediate washing with detergent and warm water is crucial, followed by treatment with 1 to 2 normal sodium hydroxide and 1:10 dilution of bleach or 4 molar guanidinium thiocyanate. There are few protease enzymes that can digest the prion proteins as well. Additionally, contaminated waste must be incinerated or autoclaved at 134 degrees Celsius using a strong basic solution, and all instruments should be cleaned and soaked in a similar solution to mitigate the risks associated with prions. Plasma is the fourth state of matter characterized by ionized gas consisting of positively charged ions and negatively charged electrons, making it a conductor of electricity. This thermal plasma can be super hot. In contrast, cold plasma, also known as non-equilibrium or non-thermal plasma, is partially ionized gas where the temperatures of the individual constituents, electrons, ions, and neutral atoms, is different, with electrons being much hotter than the heavier particles. Unlike thermal plasma, which is extremely hot, cold plasma operates at or near room temperature, making it suitable for disinfecting applications involving heat-sensitive material like living tissues and food. One effective approach is hydrogen peroxide gas plasma, which utilizes gaseous hydrogen peroxides activated by radiofrequency waves to create free radicals that inactivate prions. The steroid system are particularly useful for sterilizing heat-sensitive medical devices due to their low-temperature process, which remains below 60 degrees Celsius. The activated hydrogen peroxide breaks down prion proteins, rendering them non-infectious. And studies indicate that this sterilization method significantly reduces prion infectivity. While hydrogen peroxide gas plasma is well established, other gas plasma techniques using various gases and discharge methods have also been shown potential, particularly against scrapie prions. The slide figure on the right shows a bipolar, 2 high-voltage electrodes on each side of the grounding electrode, nitrogen gas plasma decontamination chamber. The system shows above uses 3 combined decontamination methods: low-pressure cold plasma, ultraviolet, and heat to destroy prions effectively. This device generates nitrogen gas plasma, applying short high-voltage pulses using a thyristor. Link to learning, educators' resources. Thank you.